The overall goal of the following experiment is to rapidly determine the genetic requirements for degradation of an unstable protein in Saccharomyces cerevisiae. This is achieved by fusing a reporter enzyme, required for growth under specific selective conditions, to an unstable protein of interest. Next, wild-type or mutant cells, both expressing the fusion protein, are cultured on selective medium agar plates. The plating assay provides a convenient indicator of protein stabilization on the basis of yeast cell growth. In order to biochemically confirm differences in protein abundance, wild-type and mutant cells harboring the fusion protein are lysed via a rapid and efficient protein extraction method for Western blot analysis. The results show that the abundance of an unstable protein is increased in cells with a mutation in a gene required for degradation. The converse is true for degradation-competent wild-type strains.
The main advantage of this technique over existing methods like pulse chase and cyclohexamide chase analyses is that it is less labor-intensive and time-consuming, allowing for the rapid and high-throughput determination of genetic requirements for protein degradation.
Though this method can provide insights into the degradation requirements for a range of unstable proteins, it can also be used to identify regulators of other aspects of gene expression, such as transcription and translation.
Demonstrating the procedures will be Sheldon Watts and Justin Crowder, students in my laboratory.
Prior to the assay, transform a plasmid encoding a fusion protein consisting of an unstable protein and a metabolic reporter enzyme into a mutant yeast.